Thornton Larson, and welcome to STEM Paths, where students unpack papers, study paths, and the people behind them. Our guest hosts today are Deanna and Srader, and they're going to be interviewing today Dr. Megan Frayer, currently at Yale, on her preprint in evolution of do genetic loci that cause reproductive isolation in the lab inhibit gene flow in nature? So you guys want to take it away?
Yeah, sweet. So we wanted to summarize the papers for the listeners, and this is what I got out of it. In their 2024 evolution paper titled Do Genetic Loci That Cause Reproductive Isolation in the Lab Inhibit Gene Flow in Nature, Dr. Frayer and Dr. Pacer compare lab-mapped loci for postzygotic isolation with loci that impede gene flow in nature in house mice. They found no evidence that the two sets of loci overlap beyond what could be expected by chance, and this unexpected result suggests that the genes discovered in controlled lab crosses may not be the same ones maintaining reproductive barriers in the wild, potentially due to biological differences. And rather than just methodological differences between lab and field studies, the paper urges a more nuanced interpretation of specific genetics data and calls for further empirical and theoretic work to bridge this disconnect.
Sweet. We both kind of made a summary, so I guess I'll just throw mine out there too real quick.
Just make a little addendum.
Yeah, like a little PS on there. I basically got the same thing because we read the same paper. But I said this research looked in to see whether or not genetic loci identified reproductive isolation like hybrid sterility in lab settings, and also acted as genetic barriers in wild populations. Dr. Frayer and Dr. Pacer used hybrid zones of house mice species to compare lab-mapped reproductive isolation loci with genomic regions showing reduced introgression in nature. And again, this research actually found that there was no significant overlap between the established genetic barriers within a lab setting and the regions showing reduced gene flow in the wild. It showed that lab-mapped isolation loci do not reflect the actual barriers to gene flow that natural circumstances represent, and this could maybe possibly suggest that other external evolutionary forces such as selection or behavioral isolation play more of a role in speciation and the divergence of genes in a natural setting.
Maybe possibly.
Does that sound anything close to what we were supposed to get out of that, or did we miss anything?
No, that sounded like a great summary to me.
Sweet. Yeah, and I guess we wanted to share some parts that we found really interesting about your paper, and so I'll share mine first. And so at the very end, you talk about how this mapping and these studies have inspired many researchers to call for studies that combine these approaches as a way to identify the true genetic basis of speciation. And specifically here you say, we support these endeavors. However, we encourage speciation researchers to recognize the interesting possibilities that these two strategies will point to different regions of the genome for biological reasons. And rather than purely methodological shortcomings, the presence or lack of overlap itself could be a revealing attribute, providing fresh insights into the forces that shape hybrid populations and the evolution of reproductive isolation. And so I found it really interesting that you invited the readers to zoom out a bit from trying to, or the researchers to zoom out from trying to find the true genetic basis of speciation and to consider other possible forces at play. And so I'd love to hear you expand more on that now or later.
Yeah. Well, I think for me that was one of the most kind of interesting conclusions, is because when I first did these comparisons, I thought that I was going to find a lot of overlap. And I was actually going to I was thinking I was going to use those things that overlap to say maybe these are better candidate loci than others, sort of thing. But then when I found no overlap and we started really like thinking about it and talking about it, I'm definitely a person that the methodological regions like always jump out to me more. So I think like, oh, what could I have done wrong? What could what could I have done differently that might have given us a different outcome? Have I missed any assumptions? All these sorts of things. But then when we really started thinking about like what might biologically make these different, it was kind of really interesting. We go over several different things in the paper, but for example, one of the things that I find the most interesting, I guess, is thinking of this aspect of time. So when we're doing crosses in the lab, we're usually taking two like parental individuals and crossing them together to make a first generation offspring or an F1. And so that's like one generation of hybridization, right? But when you're looking out in nature, it might be a first generation, it might be a 20th generation, it might be a 1,000th generation. And so thinking about what kinds of selective pressures might be acting differently along those timescales, I think is very interesting. And if we could be confident that we were really accurately finding the causative loci at these time points, then comparing them could inform us about those time points, which would be cool. It's kind of just rehashing what I said, but
Cool.
No, that is really, really cool. I love
Yeah.
that.
Oh, yeah.
Srader, you're on.
Sure, yeah, let's go for it. Let me steal this paper real
Yeah.
quick. I also highlighted a quote, but I've got a question afterwards. I've got a question with it. Let's see if I can find it. It's too many pieces of paper, though. Low-key.
You wrote so much.
There we
That's
go.
a
There we
great
go.
job.
Okay, so I saw one of your lines in here that kind of piqued my interest. You mentioned that laboratory studies are limited to a subset of the reproductive barriers potentially active in hybrid zones, and that genetic mapping is fairly biased toward those isolation phenotypes, and you can really only measure the ones that are strong and easy to identify. I think it's really cool that you mentioned that, because there's a lot of biases in research and people like to skip over those sometimes. But I was kind of curious if lab studies seem to be biased and could possibly miss or skew genetic data, how do you think researchers are trying to improve those mapping techniques? Or do you think that sampling of genetic material from wild populations would be more applicable in future research? Like, how would we bridge the gap between the field side of things and the lab side of things?
Yeah, great question. So a couple of thoughts. First, so some of the things that like you find in the lab that you just that could be happening in nature that you're just not going to find in the lab.
Yeah.
I guess we'll start there. Is just some of those like ecological differences. Also, so for example, if there's like some feature of their natural habitat, when you take them into the lab, you're completely removing that aspect. So you're never going to be able to understand that selection pressure. And maybe if you know what the pressure is, you can try to like recreate it in some artificial way. And that's something people do a lot and find really cool things. But you have to know what it is to recreate it, right?
So it's more about like the recreating the external stimuli to get the same results kind of thing, or?
Yeah. So what these sorts of lab crosses people do are really well geared to find are these intrinsic differences, right? Something where when you cross these mice, it doesn't matter what their environment is. They're always going to have, like, say, bad sperm. That's always going to make the males infertile. That happens in house mice, certain crosses of house mice, right? So that kind of thing you can find in the lab really easily because it's always going to happen when you do that cross versus something more specific to the environment. But there's also a portion of this that is when I mentioned things that are like easy to find, that having a mouse that is completely sterile is going to be really obvious, right? You're going to try to like get that mouse to make an F2 or whatever, and it's just not going to be able to. But if something is, I don't know, 10% less fertile, that could make a big difference quantitatively in the wild when there's other mice it's competing against and like a bigger population. But in the lab where you're maybe only going to make one, or it's only going to sire maybe like 1 or 2 litters, maybe you're not going to notice a 10%
And you're going to be
right?
controlling
So
that
that's
breeding
something else.
opportunity. Yeah.
Yeah.
So do you think it's
Yeah,
it's
so
kind of
you
like
might
the
not notice that.
combination of both the field and the lab side? Like you see more of the things that you might miss in the lab out in nature, but you can still have like a controlled setting in like it's the combination of both that really works?
Yeah,
Okay.
I definitely think so. You really need to be thinking about both. And it's, they both have their limitations because there's also, if you're just picking up random mice in the wild, you don't know who their parents are.
Yeah.
You don't know which ones are their offspring. So you're losing a lot of that control, like you said, right? So it's really good to be able to do both approaches and think about the strengths that each of them have and make sure that you're taking those into account.
Okay, cool. Thank you.
Yeah. So we wanted to know more about this paper. What was the initial idea that really got this study going, and especially the decision to test whether lab map sterility, viability loci correspond to reduced gene flow in a natural hybrid zone?
Yeah. So the thing that really got this going was as part of my my dissertation was actually looking at genomes from hybrid mice from this hybrid zone, and that work is not out yet for a number of reasons. But as part of my dissertation, I put this data set together because my intent was actually to compare these loci to interesting features that I found in the genomes that I had. And then I was kind of sitting there with this big data set that I put together of all these different papers. And I think, I forget the exact number of papers that went into that table, but I read like 100 papers or something like that. And I was like, well, is there anything else interesting that I can do with all of this data that I put together?
Yeah.
Because it was a lot of work to organize all of that. And this was sort of something that my advisor, Brett, who's the other author on the paper, that we had kind of been discussing as part of, my ongoing research, is like trying to think about how these lab data sets, which I have never generated a lab data set, but other people in the PACER lab have, versus these nature data sets, which mine would fall more into that category. And we have other people from the PACER lab, and of course, many other mouse researchers who have those sorts of data sets. We've been talking about this kind of dynamic, so that's sort of how that came about.
Quick question. I know I mentioned it in our correspondence a little bit, but you're reading all of these hundreds of papers to assemble a data set. Is that also what kind of triggered your building of the review paper you wrote?
Yeah. So that kind of the review paper that I wrote was sort of a different project, but it definitely was like foundationally started by this same. Well, let's see. I guess what I want to say, sorry. What I want to say is I applied that same sort of technique that I used to build this mouse data set to expand it over. And there are there's one gene that is in both data sets, I guess.
Sorry, that just entered my mind to ask that question because of when you read that much literature, breaking into a review paper seems like a step that makes sense. So I had to spring to there.
Yeah.
So.
What impact did you and your team hope that this paper would have, and do you feel like this comparison accomplished that?
Yeah, so I think what I was hoping was just to get other people thinking about making these comparisons, because there's a lot of really amazing speciation hybrid zone study systems where people have only lab data or only natural data. But there are more and more these systems where we have both. And so what I really wanted out of publishing this was to get other people to make these comparisons in their systems with data that they had access to that I didn't. And that worked at least a little bit. I know of at least one case where somebody, after this paper came out, had the data and directly made the comparison in a totally different system, and they actually found the same thing.
Wow.
Oh, very cool.
That there was no overlap. Yeah. Yeah. So for me, that was awesome. that was like exactly what I wanted out of this. But
Totally.
yeah.
I guess while we're talking about it, what kind of challenges did you have to face while doing all this research?
Other than the reading, obviously.
Besides the 100 papers.
Yeah. Yeah. Well, so in this particular, this was probably actually one of the easiest papers that I've written, mostly because I think the idea is very clear, at least to me. Hopefully clear to the reader, but I had a very specific sense of like, we're asking a question, and it was more let's see, it's like more of a conceptual paper, right? There's we asked a specific question, we did a specific comparison, we explored why we might find that result, but it wasn't most other things that I've worked on have had a lot of like complicated data where you just end up with, these gray areas of like, well, we found this really cool thing, but there's some caveats and we don't know quite what to do with them and that sort of thing. So, yeah. So I would say that for this, the biggest challenge was getting all of those data sets together, and actually a very small kind of technical part. But as we, as science progresses, the way we report things changes. So all of these papers that are included in this data set come from a lot of different, from a pretty broad range of years. Off the top of my head, I can't think of what the earliest one would be, but I'm pretty sure there's some that are from the 90s. And all of these papers, or many of these papers, are built across different versions of the mouse genome, mouse like genome assembly and coordinate system. So actually, one of the biggest challenges that I did get input from some other people on was like, how do I make sure that I'm actually comparing apples to apples in terms of like the positions that they call for older papers versus the newer papers, to make sure that I was all comparing the same actual spots in the genome, if that makes sense.
Yeah. You had to line them all up. That's crazy.
Do you want to ask about the pie charts?
Maybe later. That's the bigger one. I was going to go off of, like, you kind of just said, compared to other studies you've worked on, this was a little easier. Like, it was a little nicer to work with. I don't know how much other research you've done or other studies, but compared to other ones you've been on, what is this saying? Oh my God. Compared to I'm just going to go off the dome. Compared to other research you've worked on, like, have you done a lot of field stuff too? Like, how different was this one to work with compared to other studies you've been on?
Yeah, so I think let's see. I mostly am computational. I haven't done a lot of like field studies. I've done some field work, but it's mostly been like collecting, not necessarily trying to study something that's actively happening in the field, which is much, much harder. But yeah, I think what's kind of different about this paper is it's really in the vein of meta-analysis, right, where you're taking other people's data and trying to learn something new from it and then ask a new question in a new way, sort of thing. When you have to make your own data, it's a lot. There's a lot of challenges that come with generating data. Yeah. Whether there's all of the technical aspects. Do a lot of genome sequencing, and there's lots of technical hurdles of at all of the different stages of extracting your DNA and all of that sort of stuff. Then you get it back, and there's always something that's not what you think it's going to be. And that sort of thing. So knowing, I guess, having these studies coming from published papers, I like knowing what they all were going in, even though there were, like I said, the technical challenges of making sure they're all in the same coordinates and that sort of thing. At least you know what the data is going to be when you sit down with it.
That's kind of nice. That's kind of nice.
Yeah. So we already talked about it a little bit. Dr. Larson mentioned the review paper that you wrote, the Molecular Evolutionary Basis of Species Formation Revisited, which we both found super interesting. And before this, we were specifically talking about the very cool pie charts that you had in that paper that were showing proportions of hybrid incompatibilities, as well as early and late life lethality. And we thought that the visuals were very informative. And could you expand more on that research? And you said it was kind of born out of that initial paper. And so where's it going?
So I guess for this review, it sort of came about in a little bit of a different way. Just to be clear, I had been thinking about these things because of the other paper that we have been discussing, but I kind of got invited into this group that consisted of the two last authors, basically, who were thinking about doing this review. So that's sort of how that came about. But the idea was that there were a bunch of papers around 2009, 2010, where people tried to put together these lists of all the known incompatibility genes. But there haven't really been any updates since. There ended up being another update that came out this summer or this spring, I guess. I think we cite it in our review that we were doing it kind of at the same time unknowingly. We thought that there was time for an update. There are a lot more genes now. I think the main paper that we cite in that our title is actually a reference to, a paper called The Molecular Evolutionary Basis of Species Formation. That paper had, I think, like 9 or 10 genes in it, and our table has like 100.
Oh my gosh.
So clearly, yeah. So clearly it was time for an update, but it was also really interesting to think about, I guess, how the things included in that table have really changed. I think coming back to the pie charts that you mentioned, the exact numbers escape me, but I think that first table had like 3 or 4 species that were included. And now we have tons of species that kind of span the tree in a much more, still not comprehensive way, but covering a lot more branches of the tree at the very least. So it's just giving us more insight into, how things are different in different parts of the tree of life.
Cool.
That's really cool.
Thank you for sharing.
Did you notice, I know that's not the paper that we're actually talking about today, but I was kind of curious on that. Did you notice any difficulties working with classic or not classic model organisms? I know you guys spread out the taxa that you worked with a lot for the hybrid incompatibilities. Was there any crazy stuff you had to deal with the new stuff?
Yeah, so for everything in this paper, we're really just collating existing studies. So we didn't find any new genes or anything like that. We're just kind of putting together all the things that people have found and thinking about kind of the patterns that they find. But there were definitely some interesting ones. I think I did not really appreciate how much work has been done in a lot of the crop plants. Because I think that a lot of the papers, it was kind of a fun challenge because it was pretty easy for me to make a first draft list of all of the genes that people have thought about really in a speciation context, because I think about speciation and I keep up with that literature. But then there's all of these really interesting genes that people have found in the context of crop breeding that are incompatibility genes, right? They're things that are preventing different lines from being able to be crossed together, but they write about them and think about them in a way that's more relevant to crop breeding versus the way that I think about it. So so trying to understand some of the differences in the way they're talking about things and describing things, and that was challenging but kind of fun.
You mentioned something so funny to me because it was, I think 2 years ago, I was invited to an agricultural genome conference by some professors we had here at the time. I didn't even know this world existed, right? Because I'm so focused on my phylogenomic questions and things like that. So when I learned how big the ag field is in terms of genetics and everything, it was like, whoa, how did I completely miss this? And it's funny in the sense of how you can completely miss some field of research that actually could help your understanding
Yeah.
in another area.
Yeah.
Yeah, definitely.
What was the writing process like for this paper, and any tips for making a negative result, like no overlap beyond chance, maximally informative for the field?
Yeah. So the writing process was kind of interesting because at first we were, well, like I said, I kind of did the, made this, or I didn't assemble this data set with this paper in mind, right? I was doing it for something else, and then I thought, how else could I use this data? And at first we were thinking of trying to do what you would consider as like a more formal meta-analysis. But we couldn't really, we were kind of struggling with doing that because of all the different data types. Some of the statistics that you might want to use just weren't really going to work. So that's when we kind of switched to going for this more conceptual sort of style of paper, which I had never written anything like that before. But it was sort of, once we kind of had the idea that that's what we were going to do, it was kind of fun because there's no rules about format, right? Like most of our papers that we write, you have your introduction, you have your results, your methods, you know, your discussion, and there's kind of prescribed things that go in each of those categories. But this paper, we could be a little more, think about the structure that made sense in the order we want to talk about things, which was kind of fun. Yeah, was there a second part of that question? Sorry.
Just like how to make negative results informative and mean something, I guess, to the public.
Yeah. Yeah, I think that the best thing to do, I guess, is just to not, you know, I could have just said like, oh, there's no overlap, that's not interesting, and like walked away from it. But I guess the main thing is just to really think about, you know, why is there no overlap? Is it a technical reason, or could there be interesting biological reasons? And we couldn't distinguish between those things, but I think that there are enough sort of interesting options that we could like discuss them all. Which is what we did in the paper. Yeah. So I think, just because the result is negative doesn't necessarily have to mean it isn't interesting. So just think about, you know, the reasons you might have that negative result, and if any of those are informative too.
And you mentioned that there is another researcher that did a similar study after you published that and found a similar discrepancy. So that's definitely meaningful.
Yeah.
Do you enjoy writing as part of your research? And what's your favorite way to share findings in an academic to an academic audience versus the general public?
Yeah, I do enjoy writing mostly. I think, I think a lot of researchers have the experience of when you get to the end of a project, you know it's ready to go when you're just kind of sick of looking at it. And I definitely have been there on projects before, but for the most part, I do actually enjoy writing because it's nice to kind of, for me at least, I think I really enjoy the part of it that is the synthesis, right? Like thinking about what you actually found, how it fits into the context of what we know, and writing can kind of help you think through all of that. So, yeah, I do. I do enjoy that. But
very cool. Kind of the putting it all together part of it is fun for you. That's sweet. I know you already said you're from Yale. How did you get to that? Like, how did you go from wherever you started all the way to being a doctorate at Yale? That's really cool.
Yeah. Well, so I started out doing undergraduate research. I got my bachelor's degree at Michigan State, and I worked in a lab where I was mostly just rearing fruit flies. And so these were fruited fruit flies. They're Ragolitis genus. And so it's really fun. You would go out and pick fruit off of trees. They live in all sorts of different kinds of trees. I was mostly focused on these ones that live in junipers. And so we'd go and pick juniper berries off the tree, bring them back to the lab, rear out the flies, and then sequence their mitochondrial DNA.
Oh, that's cool.
And yeah. And so that's sort of how I got into research, and I really enjoyed it. And so I kept going. I got my PhD at the University of Wisconsin, and I was there for a long time working on the house mice in the hybrid zone. And then now I'm at Yale, but I'm a postdoc at Yale, so I'm working in somebody else's lab. I have a fellowship, and I'm continuing to think about hybrid zones, but now in these monkey flowers.
The mimulus ones you were talking about?
Yes,
very cool.
mimulus, yeah.
So you've always been like kind of focused on the genetic side of things? That's kind of really piqued your interest?
Yeah, I really like thinking about, so I guess I do have always done genetics, but I really like thinking about evolutionary biology.
Oh, okay.
That's more of my, yeah. But the intersection of evolutionary biology and genetics is sort of where I have stayed.
Very cool.
Definitely. What do you most enjoy about your role right now?
Yeah, so I think being a postdoc is a little stressful because it's not a long-term position. You have to find another job eventually. But that being said, it's a very like free and flexible sort of position. Like I'm just doing research. I don't have to like teaching obligations or things like that. And I just get to spend all of my time thinking about my research, doing my research, and that's really fun.
What advice would you give to students interested in your field?
Yeah, so I would say definitely just if you're an undergraduate, like try to get involved in undergraduate research. And even if you, I think sometimes students are worried that like the lab that they can get a job in isn't necessarily doing what they want to do forever, but like people jump all around and work on different things. And so just getting some experience and understanding, how you read a paper, how you write a paper, how you do research in a lab and lab safety, and like all of these things are all like good transferable skills. So just getting some experience and seeing if it's something that you're actually that you actually enjoy doing.
Totally, yeah. Because you never know what in that field you might be able to take over to another one and have a revelation, an unexpected revelation. Definitely.
Yeah, absolutely.
What has been the most rewarding aspect regarding the scientific aspect of just science in general, doing science?
Yeah, well, that's a great question. I think I really just love, learning things that people haven't known before.
Right.
Right. And
the exploration.
Yes. Yeah. When you can, even if it doesn't seem like, an earth-shattering question of like, what's the, or how does like this population, something I'm thinking about right now, how does this population of wildflowers actually relate to this other population of wildflowers? Like, even if it's, not going to change the world, maybe to answer that question, just being like the first person who, is looking at these genomes and going to figure it out is just kind of fun.
Yeah, super special.
That's pretty cool. I was kind of curious because you said that, the postdoctorate isn't like a super long-term thing. Do you have any idea what you want to do after that? Like, have you started looking at stuff already?
Yeah. So you may or may not have heard that the academic job market is pretty, can be brutal.
Yeah, a little bit.
So that's where I'm headed, but not quite there yet. So.
So you're thinking like professor role?
Yeah.
Okay. Do you want to stick at Yale or just?
I'm sure I will go somewhere else, but I don't know where.
Okay.
Yeah. It tends to be the way this works is that postdocs apply to like dozens of jobs all over the country or even outside of the country. And then if you're lucky, you get one.
Sure. Yeah. And going off of that too, what are some of the biggest challenges you think are facing scientists and researchers today?
Oh boy.
Boy.
Yeah. Well, there's plenty, but I think, I think, well, so, sorry. There's so many things come to mind, but I definitely think, the relationship between like the public and science right now is pretty,
Not great.
is at a different stage than it has been in the past. And I think we all have to figure out how to navigate that. And,
Yeah.
There's our actual practical, like how am I going to continue to get paid kind of questions, but there's also, how do you relate your research or just science in general to people that are now coming to science with more baggage and ideas of what your answers should be and that sort of thing. And I think everybody has to face that at some level.
Have you had to change like your presenting strategies or like the way you kind of propose research recently with all the changes that have been going on? Like, have you noticed any differences?
Yeah, so I'm in kind of a very fortunate spot right at this moment where I am on this fellowship and I cannot apply for I can't have any other funding while I'm on this fellowship. So I'm not actively applying for things right now. But if I was applying for things, I would definitely probably be talking about things differently than I have in the past, which is unfortunate, but yeah.
And that disconnection between scientists and the public, do you feel like it's more because of the education, or maybe scientists need to adjust the way that they bring data to the world and maybe make it more understandable, relatable in some way?
Yeah, I think that has always been a struggle. I think especially the more like specific your research is, if you are like running clinical trials for cancer drugs, right? You don't have to explain to anybody why that's important.
Right.
Everybody knows that that's important. But if you're doing things that are more, like I'm working on these populations of wildflowers, it's people need a little more convincing of why that matters. And I think that's something that has always been a struggle, and it's just going to be even more of a struggle now. Oops.
Yeah, I think
I might have just lost my. Okay.
Uh-oh.
Can you still hear me?
Yep, we can hear you.
Okay.
Can you hear us?
I just lost one headphone.
Uh-oh.
Yeah, I think a lot of the some of the interesting things about that, we try to convey it in early levels of science courses about the differences between applied science versus basic science. Applied, you're usually trying to solve a problem that people can relate to, whereas basic science, it can sometimes be described as knowledge for knowledge's sake.
Just asking why,
which to any science person, they're like, this is awesome.
Yeah.
But a lot of people are like, why would I spend money on that? Why would I want to spend money on studying, I don't know, think of your favorite thing in a county in the world.
Why should I care about the genetics of a tiny beetle out in the desert that doesn't affect me in any way? Because it's cool.
Because it's cool. Right, exactly. Oh, man. We've got 3 more questions for you just to kind of wrap things up a little bit. So if you guys want to start those 3 questions there.
Okay, what's one piece of advice you'd give your undergrad self?
Okay, yeah. A piece of advice, let's see.
You spoke about it a little bit earlier.
Yeah, I would definitely say that I would advise like all undergrads who are interested in science to just get involved with research. And I think like just keep going forward. Like science is really challenging, but it's also really rewarding. So just because, the first thing you do doesn't work out, like that's okay. A lot of things you do won't work out. And that's sort of one of the joys, right, is finding how things work and how things don't.
I think that's the underappreciated thing that we learn better as we get a little older, is the learning attributes of failure.
Yes.
Of having to take those steps back and reassess and get back up on the horse and keep going. I think we understand that better as we get a little older than we do when we're younger.
Definitely.
Oh, right.
That's fun.
So I guess if you weren't doing this, what would you want to be doing right now?
Like instead of being a scientist?
Yeah.
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 Any job in the world.
Yeah. Well, when I was a kid, I wanted to be a zookeeper. And I worked at the zoo for a couple of summers. I was pretty serious about that. And
Awesome.
then I got kind of swayed that I enjoyed doing research more. But they do plenty of research at zoos too. But
Zookeeper would be really cool.
Cool answer. Yeah.
You always have a plan B.
yeah.
And so our final question is, what's one thing you wish more people understood about science or scientists?
Yeah. Well, I would say that I think, especially in light of the things we were just discussing, that I think it'd be great if more people understood that scientists are just people, like out here doing our jobs, trying to do our best. And sometimes scientists make mistakes, but at the same time, we're just, normal everyday citizens, just doing a job. So, yeah.
All right. With that, I want to thank our guest today, Dr. Megan Frayer, currently postdocing at Yale, and our guest host for doing such a great job, Deanna and Srader. This has been STEM Paths. I'm Dr. Thornton Larson, your wrangler. And have a good one. Thank you, everybody, for making the time. I really appreciate it.
Thank you.
Thank you. It was super nice to meet you.